Good morning, our students. How do you do? Today we have the first lecture of practical medical parasitology concerning the flax or Nematoda worms in the helminth worms. Now, there is specific criteria and characteristic criteria or features concerning to the general in a whole. Now, it is a general term for a parasitic worm and comprises a group of multicellular organisms known as the flatworm and the roundworms that are found worldwide and some of the most common parasites in poor, less developed countries in the tropical and subtropical regions, causing serious disease due to migration in the body. Worms are relatively large and have well-developed organ system and can be dioecious, different sexes, or have both sexes hermaphrodites in the same worm. Here, this criteria is different from that found in the protozoal parasite that we studied in the first course. Here, multicellular, there is organs and organelles found in this group of parasites, the worm. The life cycle here also is different from protozoa parasite. The life cycle of many hermaphrodites is complex and may involve more than one host. Definitive host here is the host in which the sexually mature adult helminth lives. In order for the parasitic to continue its life cycle, the larval developmental stages forms of the parasite may need to be passed from one intermediate host to another, so there is more than one intermediate host. About Nematoda group or class called flax. This parasite can parasitize members of all the vertebrate classes, but most commonly parasitize fish, frogs, and turtles animals. They also parasitize human, domestic animal, and invertebrates such as mollusks and crustacea like fish, crab, and so on. So, some are external parasites, this means ectoparasite. Some attach themselves to internal organ, called endoparasite. Others are semi-external, attaching themselves to the lining of the mouth, to the gills of fish, or to the cloaca, the end of the digestive tract here. Some attack a single host while others require two or more hosts. Now here can show you the specific criteria concerning flax worm. Here the criteria concern flax only group of parasite. They are leaf-like, resemble the leaves of a plant in thickness and shape, and ovoid or oval in shape, and non-segmented. This is good criteria, non-segmented. The body here flatted, non-segmented. They are provided with suckers for attachment purposes. One anteriorly located, called sucker, and B or second is posteriorly located ventral sucker or called acetabulum for the attachment purposes. They are hermaphroditic, both sexes in one organ. Some of them hermaphroditic, whereas the blood group system species is separate in sex, dioecious, not hermaphroditic. All require body of water to complete its life cycle, because the snail, the fish, crayfish, all these intermediate hosts be found in the aquatic environment, so it be needed to be used to complete the life cycle of the parasite, to be passed to complete the parasite life cycle. Now, general rule in the classification of a flax or Nematoda: all flax are leaf-like except for the cystosoma, rounded in cross section, not like this. It's different. This group from this class is different from other. It's rounded in the cross section, not leaf-like. Now, other criteria: all flax are hermaphroditic except for the cystosoma 6 be separated. All require 2 intermediate hosts, except for the cystosoma also having 1 intermediate host, which is nil. Now, the effective stage for all this group of trematode parasite is metacercaria, insisted metacercaria. So, with the exception of all the cystosoma species, it is a cercaria that penetrates the skin through swimming, skin of a human being. Now, the mode of transmission is by the ingestion of the infective form, except for the cystosoma by skin penetration. The cercaria infected so of the cystosoma species can penetrate the skin to cause disease. Now, all eggs are operculated, having cover like, except for cystosoma also there is no cover, no operculum, but equipped with spines, lateral and terminal spine. About this slide, about the host requirement for this parasite. Now, first, one definitive host harbor the adult stage of the parasite in human. Second, there is 2 intermediate hosts. The first intermediate host harbor the early larva stage of the parasite in snail in water, and second intermediate host that harbor the stage of infective stage to human in fishes, which is metacercaria. Now, about its classification according to its habitat. Now, the first group is lung flax, like as example Paragonimus westermani, that parasitize the lung, dwell the lung of a human being. And second group is intestinal flax or trematode. An example here, Phaeolopsis buscae, Heterophis, and Metagonimus yakagawa. And any species be found but not listed here in this lecture, but we consider them those who with medical importance. Now, third group is liver flax. As example, here 2 example, Fasciole hepatica, the lecture of today, and the Clonosis sinensis, also the lecture of today. Okay, fourth group of blood flax, an example, Schistosoma species. The first Schistosoma japonica, second Schistosoma masoni, and the Schistosoma hematobium. All these represent or can cause illness, serious illness to human being. Now, this slide can show you the first species of sheep liver flax as a common name of a blood flax causing fascioliasis. This flax found in herbivores that graze in wet pastures where intermediate snail hosts are found. Fasciola hepatica egg shed from the infected primary host and mature into the embryonated form in the environment. The embryo hatch and release the motile miracidium, the first intermediate host larva stage, miracidium, that seek out and penetrate the tissue of the first intermediate host is a snail. Now, the cercaria be out or exiled or discharged from the first intermediate host snail to be produced in the cell and then released into the environment, and can insist to produce the infective stage, which is metacercaria. Man is often infected by eating wild watercress on which the metacercaria have collected or attended. After being ingested, the metacercaria pass through the dural wall and penetrate the liver capsule, so the adult flax develop and live in the bile ducts, causing damage to the biliary tract and adjacent liver tissue. The eggs are passed with the bile into feces to continue the life. Now here this slide can show you the adult worm of Fasciola hepatica. Here the picture can show you the internal structure and the suckers and the ceca and the reproductive organs, testes and uterus and collet or twisted ovary and vitelline gland and spines that cube the body for test purposes. All these criteria can be presented and depicted here. About second stage, which is the egg of fascia hepatica. Ova are thin shelled, as you see, ellipsoid, oval in shape, bile stained, guanine colored, yellow colored, with an operculum which is inconspicuous. It's been difficult to see. It's found, but you need to consider to give the good observation here. Okay. Now about the life cycle and the transmission. How can be completed and how can be transmitted to human being. Now the life cycle, as we know, it's very difficult because it needs more than one intermediate host. The adult worms inhabit the liver or bile duct of the definitive host, human or man, and there are or where they lay many eggs which are deposited into the environment in feces. They are immature when passed, very important criteria. It be immature when be passed out. If they are passed into the water, they become mature in 9 to 15 days, 2 weeks approximately, to be matured at the optimal temperature of 22 to 25 degrees. This shape can show you in the human the adult flux found in liver, biliary duct, and can give egg, the egg having undeveloped an embryonated egg in feces be discharged out. The embryonated egg here will be formed, not unembryonated but embryonated, and having the miracidium. The miracidium be excised out and to seek for the snail as a first intermediate host, and to be developed to sporocyst, then to redia, and then to cercaria. This cercaria can leave the first intermediate host snail and free living, and can enter or be insisted on the water crust or plant, aquatic plant, to be known as infective stage for human being or other animals. Now, if ingested by the human being, the metacercaria can be exist in the duodenum and attach to mucosa of small intestine, and then penetrate the wall of GI, then perineal, then to liver parenchyma, then to be matured in the adult in the biliary tract or biliary duct. Can harbor the adult form or mature form of this. Now the life cycle of this parasite here can be written, and can you see how can the egg be transformed to many stages to be developed to form cercaria, and then the cercariaries from the and insist as a metacercaria on aquatic vegetation or other surfaces. Mammals acquire the infection by eating vegetation containing metacercaria. A human can become infected by ingesting metacercaria containing freshwater plants, and especially watercress. After ingestion of metacercaria sits in the duodenum, and then complete its life cycle and development to reach the liver to be developed that or within it, especially in the large biliary duct. In human maturation from metacercaria to adult flux takes approximately 3 to 4 months. The adult flux fascia hepatica reside in the large biliary duct of the mammalian host. So fascia hepatica affect various animal species, mostly herbivorous animals, not just the human being. Now, how can we diagnose this parasite in laboratory? Fasciolosis, we mean fascia hepatica, have been diagnosed by observing the egg during fecal examination. The first. Second, by parasitic specific antibody detection in a variety of immunodiagnostic assays. So serologically can be diagnosed immunologically. Now by radiological method, laparoscopy. Fourth, data history is also helpful part of the investigation outbreak. Here we must concern the case history, eating program, endemic region. All of these data can help to diagnose the persuaded or detected parasite. So the definitive diagnosis is made by observing the ova and feces. Since the flags are very prophylactic, any significant infection will be easily picked up, while identification cannot be made from the size of the ova. Clinical information and the source of infection may help to provide a diagnosis. So you know, our student, so you know there is many similarity found between the eggs of the parasite, and we cannot depend only on the size of the or shape size of the egg to differentiate between the species. We must depend on other criteria to give accurate diagnosis. Now, about second species that related to the blood flux or trematode, called Clonorchis sinensis. Its common name is called Chinese or Orca oriental liver flag. Adult flag live in the liver and produce egg that contain miracidia that are released and penetrate the snail to eventually produce cercaria that affect the muscle of fish that will insist. Now the human are exposed when they ingest the cyst in raw or poorly cooked fish. Once exposed, the metacercaria will mature into adult and migrate to the liver and affect the gallbladder and bile ducts that can cause blockage of the duct, biliary duct, gallbladder. And Clonorchis sinensis, also known as the Chinese or oriental liver flag, is a narrow, elongate liver flag found in Far East, mainly Japan, Korea, China, Taiwan, and Vietnam. Now this or all these members of this group are parasites of the fish-eating mammals, not just the human being. Particularly in Asia and Europe, man is the definitive host, and water, snail, and fish are the intermediate host. Infection can be easily avoided by man not eating raw fish, since there is the only way that infection can be passed on. The Clonorchis sinensis parasitize the biliary duct in a human who become infected by eating raw or undercooked fish. Dogs and cats and other carnivore animals are the most important behavioral hosts can harboring this. Now about its morphology. The morphology here depicted in the 4 pictures in this slide. The shape is lanceolate shaped, this parasite, and appear translucent and brown in color. They are all hermaphroditic. It's mean keeping in common with other flags. They possess 2 suckers, also having anterior and posterior suckers. Okay, the ova of Clonorchis sinensis contains fully developed miracidia, not undeveloped, but developed miracidia, which differ from the first species, especially hepatica, and possesses prominent operculum. A prominent operculum, not difficult to see, not inconspicuous growing. Now there is a shoulder found beneath the operculum of the egg. So flask-shaped egg. Very criteria here can represent this group or species, and are operculate. They are bile stained and measure 29 micrometer by 16 micrometer in wet mount. They are transparent and you can quite easy see the anatomy, internal structure. Now there can be up to 6,000 worms present and a daily egg output of 1,000 egg per microliter of bile or 600 per gram of feces as a dose of to can cause infection. Now about its egg, the second stage ova or egg of this parasite Glaucocyansis, showing the prominent operculum shoulder in which make identifying this nematode easy. They are described as a flask shaped, bile-stained. The cercaria possess other larva stage cercaria, pulses, eye spot. The infiltration and cytogenous gland are also well developed. Saline smear showing the characteristic flag shape of the Glaucocyansis ova. They are bile-stained with a smooth outer coat, as present here in this two figure or shapes beneath. Can show the life cycle and transmission route. The life cycle of this parasite changes the flag. There are, the parasites require the involvement of two intermediate hosts, freshwater and snail, freshwater, snail and fish to complete its life cycle. Embryonated eggs are discharged in the bile duct and in the stool. Well, the egg ingested by the suitable snail as a first intermediate host. Okay, each egg releases a miracidia which go through several developmental stages like sporocyst, lydia and cercaria. The cercaria are released from the snail and after a short period of free swimming time in water they come in contact and penetrate the flesh of a freshwater fish, so as a second intermediate host, where they insist to be encysted. This means metacercaria found in the vessel, the muzzle of the second intermediate host, which is fish. Okay, infection of a human occurs by ingestion of undercooked, salted, pickled or smoked freshwater fish. After ingestion, the metacercaria exists in the duodenum and ascend the biliary tract through the ampulla of Vater. Maturation takes approximately 1 month. The adult flags reside in a small and medium-sized biliary duct, not in large biliary duct, but in small and medium-sized biliary duct. In addition to human, carnivorous animal can serve as a reservoir host, as present here in this figure beneath. The embryonated in the first species, but here the embryonated egg be discharged. Now injected by snail. Now miracinous process lydia to give cercaria free swimming. Then be enter penetration to the visual of the fish as a secondary host to be assisted as a metacercaria in the fish. Now ingested by human. Now exist in the duodenum and then be to complete its life cycle, migrate to the bile duct through liver to bile duct, then as adult form found. Now about its pathogenicity or pathogenesis. Many millions of people become infected every year, but only a minority suffer from any illness. The pathology is related to the number. Okay, the pathology here, listen my student, the pathology depends on what? On the number of the parasite present. It's the first. So, in light infection, it is up to 15 eggs or more are usually asymptomatic. There is no symptom. The patient, the affected individual cannot be symptomized because the number of egg is 15 egg or more. Sorry. But at heavy infection of 500 or more egg may cause serious illness. So, in the disease symptomizes the acute infection, at acute infection may be characterized by fever, diarrhea, epigastric pain, enlargement and tenderness of the liver, and sometimes jaundice. The invasion by this worm in the gallbladder may cause cholecystitis due to the flux becoming impacted in the common bile duct. Okay, now about its laboratory diagnosis. In laboratories, how can we diagnose it? You know, the definitive diagnosis is made by the observing the characteristic of feces following an iodine staining formulated concentration method of the feces or fecal smear or sample, or other technique, other way to detect it from the non-aspirates when there is a complete obstructive jaundice. Other method from the intero test. These three methods listed here can be used to detect this parasite infection. And